Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing Kerala SET Biotechnology question paper conducted in the year July 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of Kerala SET Biotechnology been answered, you can go to my channel Omni Bioentrance. In the channel, select playlist. In the playlist, see for Kerala SET Biotechnology and you will find them there, or Kerala SET exams. Then next one. What percentage of the human genome was estimated to be protein coding genes? It is less than 5%. Then next one. If the mean and standard deviation of the series A and B are as XA, XB is given, sigma A square, sigma B square is given, which of the two series is more consistent? So the more consistent will be the one with lower coefficient of variance. So now calculate the coefficient variance. In the last January 2024 paper, I told you the formula for coefficient of variance. So coefficient variance, standard deviation by mean into 100. Substitute the values for series A. Series A standard deviation is 25. By mean is 15 into 100, so 166.67%. Similarly, series B you will get 80%. So series with lower CV is more consistent, so it is series B. So the answer is option C. Then next one. The computational methodology that tries to find the best matching between two molecules, a receptor and a ligand, is molecular docking. Answer is option D. Then the operating system and the other processes are protected from being modified by an already running process because every address generated by the CPU is being checked against the relocation and limit registers. Answer is option A. What is the main function of heterocyst? Heterocysts are found in cyanobacteria and its main function is nitrogen fixation. Answer is C. Identify the incorrect statement about episome. So episomes are DNA molecules that can replicate independently of the cell's chromosome. They get integrated into the bacterial genome. Now what is the difference between plasmid and episome is that plasmid do not integrate into the genome, whereas episomes can integrate into the genome. So they occur in bacteria and prokaryotes, that is correct. In bacteria you can see episomes, as well as in eukaryotes they occur in yeast. Then they can't replicate independently. They have asked for the incorrect statement, so answer is option B. They can replicate independently. They can integrate into host chromosome. Yes, they can replicate as a part of the host chromosome. That's also correct. Then blood agar is an enrichment medium. Answer is option A. Sorry, it is an enriched indicator, not a medium. It is an enriched indicator. Answer is option B. Then Macintosh fills jar is used for anaerobic incubation. Answer is option D. Volantine granule consists of polymetaphosphate. They are found in bacteria. Then next one. Which of the following is not sterilized by moist heat methods? It is liquid paraffin. This is because moist heat cannot penetrate liquid paraffin. So it requires alternate sterilization methodology such as dry heat sterilization, filtration, or using chemical sterilization. So answer is C. Then the antibacterial activity of a disinfectant is determined by Riddle-Walker test. Answer is option C, also known as phenol coefficient test. Then ELK test, it detects diphtheria toxin production by Corynebacterium diphtheriae. DIC test is assess immunity to scarlet fever. And germ tube test, it identifies Candida albicans based on germ tube formation. So answer here is option C. Then immunity obtained by serum therapy is an example of. So it's an example of artificial passive. So it involves, in artificial passive immunity, it involves administering antibodies from an immune individual to provide protection against specific infections or diseases in an infected individual. So you take antibodies from an immune individual, inject it into the diseased individual to provide a temporary protection against the infection or the disease. That is artificial passive. So immunity obtained by serum therapy is an example of artificial passive immunity. Natural passive is transfer of immunity from one individual to another, that is from mother to the fetus through placenta or breast milk. Natural active is refers to body's natural defense against infectious diseases. Then artificial active is used through vaccination, that is you stimulate the immune system to produce specific immune response, that is artificial active. Then which of the following immunoglobulin classes can cross the placenta? It is IgG can cross the placenta. Then identify the correct statement about IgM. It is present in mucous linings. So it is IgA that is present in mucous lining and offers local immunity. So it is not IgM. You have to find the correct statement about IgM. Then it is the immunoglobulin class present in largest concentration in serum. No, it is IgG that is present. Then it is the largest immunoglobulin molecule. Yes, it is pentameric in structure. So C is the answer. It has critical role in the pathogenesis of atopy. Atopy means allergic reaction. So allergic reaction IgE is involved, not IgM. Then Audin procedure is, it is a single diffusion in one dimension. Answer is option A. In this method, what they do is antibody is mixed with agar gel in a test tube and it is considered the stationary phase. Then you add antigen solution into this. The antigen then diffuses downward through the agar gel, forming a line of precipitation or lines of precipitation. So the number of bands formed indicate the number of different antigens present. So Audin procedure, that is single diffusion in one dimension, is the correct answer. Then which of the following is single radial immunodiffusion? It is McNeely technique. Answer is option D. Then which of the following is a dimeric molecule? It is IgA that is dimeric. Example of non-professional antigen presenting cell, thymic epithelial cell. A itself is the answer. Dendritic cell, B cell, macrophages are professional antigen presenting cells. The disease characterized by development of autoantibodies against TSH is Graves' disease. DiGeorge syndrome is reduction in T cell production. Addison disease is autoimmune destruction of adrenal cortex. Crohn's disease is inflammatory bowel disease that affects the lining of intestinal tracts or digestive tract, that is Crohn's disease. An example of antagonistic drug. Now before coming to what is antagonistic, you should know what is agonistic. Agonist. Agonist drugs, they are substances that bind to the receptor and produce a biological response. Whereas antagonistic, what they do is they bind to the receptor and block or interfere with the or reverse the action of the agonist drug. That is what antagonist do. So an example of antagonistic drug is naltrexone. Answer is option C. ABD, that is heroin, methadone, oxycodone, they are opioid agonist. They stimulate opioid receptors. Whereas this naltrexone, it is an block the opioid receptors in the brain. So C is the answer. So it is an antagonistic drug. Then hepatitis B vaccine is a recombinant vaccine. Answer is C. HAT medium is used in hybridoma technology. A. Who among the following is known as father of pharmacogenomics? It is Arno Motulsky. Answer is D. Which of the following is produced via recombinant DNA technology in Chinese hamster ovary cells? It is ATPase, also known as tissue plasminogen activator, and is used to treat ischemic strokes, myocardial infarction. Another question on ATPase was asked on January 2024 question paper. Then next one, maximum capacity of lambda phage vector is 24 kb for lambda replacement vector. That's correct. Then SPI selection of recombinant lambda phage vectors have which of the features? So SPI selection, lambda phages, they are normally sensitive to P2 interference, that is SPI, and they cannot infect E. coli cells that already have this P2. Now this SPI phenotype, it is carried on the stuffer fragment of the lambda phage insertion vector. Now when you remove the stuffer fragment in the insertion vector and insert foreign DNA fragment in place of this stuffer fragment, the phage become insensitive to P2. That means the recombinant phage, it can grow on an E. coli host carrying the P2 prophage, whereas the non-recombinants cannot grow. Okay, so that is P2 selection. Then presence of chi site. Which of the following features is presence of chi site? So chi site in SPI DNA sequence recognized by RECA protein and is involved in homologous recombination. So the answer here is option C. Presence of red gene, it is involved in lambda phage recombination but not related to SPI selection. In presence of GAM gene, it inhibits REC-BCD nucleases not specific to SPI selection. Then absence of chi site means it prevents SPI selection. Then which of the following is true about C1 repressor protein? It is the only. I told you whenever this only comes, mostly about 90% or 95% of the cases that statement is going to be wrong. It is the only lambda protein synthesized during lytic cycle. No, CRO, REX-A, REX-B are also synthesized. If the C1 repressor protein is inactivated, lambda will form clear plaques. That's correct. C1 repressor protein prevents lytic growth. Okay, C1 repressor protein, it prevents lytic growth. So blocking the expression of the lytic promoter PL and PR, lytic cycle proceeds. Oh, sorry. C1 repressor protein prevents lytic growth by blocking the expression of lytic promoters PL and PR. Okay, C1 repressor protein, that is what it normally does. But when you inactivate the repressor, then lytic state proceeds. So it will form clear plaques. So answer is option B. If C1 repressor protein is inactivated, lambda will form turbid plaques. Not turbid, it is clear plaques. High frequency lysogenization is prevented by C1 repressor protein. It is facilitated by C1 repressor protein. So D is also not correct. The vector which has the highest capacity. So out of this BACMID, PHAGMID, PHASMID, you have COSMID. COSMID is the one with the highest capacity. Then heterohyphocomers are pair of restriction enzymes differing in methylation sensitivity. C. On same recognition site but digest at different position is neo-isoisomers that they have asked in January 2024 question paper. Different recognition site but produce same termini, they are isocodamers. So here answer is option C. Identify the statement true about topocloning. So topocloning, also known as topoisomerase mediated cloning, it is a rapid cloning method and it uses topoisomerase 1 to join DNA fragments. So in topocloning you need not go for DNA ligase and ATP reactions and it is fast and efficient cloning method. So complete ligation in 5 minutes at 37 degrees Celsius. Answer is B. A. Requires 5' phosphate for phosphodiester bond formation, need not. Topocloning is not site specific, it is site specific reaction. Protocol requires 16 degree water bath for incubation, not 16 degree, it is 37 degrees. So answer is option A. Then next one. Which among the following is true about nucleic acid hybridization? Excess NaCl, decreased stringency. So this is about high stringency and low stringency condition. Now what is this stringency? The stringency of nucleic acid hybridization, it is determined by temperature and salt concentration in the hybridization buffer. So high stringency condition means they allow hybridization between the nucleic acid and the probe to the region which are highly complementary, or I can say between highly similar nucleic acid sequences. Okay, there you have got a DNA sequence, a probe which is complementary to this DNA sequence. The probe will bind randomly, but in high stringency condition it will bind to complementary basis only, not to random sites in the nucleic acid. So high stringency condition include high temperature, low salt concentration. Low stringency condition means the probe binds to unrelated targets and it uses low temperature and high salt concentration. Now see the options. Excess NaCl decreases stringency. Yes. Low salt concentration. No. High salt concentration, it decreases stringency. Yes, it is low stringency condition. A is the answer. Excess NaCl destabilizes hydrogen bond. Low salt concentration only destabilizes hydrogen bonds, not excess NaCl. High temperature decreases stringency. High temperature means high stringency, not decreased stringency. So C is also not correct. Formamide stabilizes hydrogen bond. Formamide destabilizes hydrogen bond in nucleic acid hybridization. So A is the right answer. Labeled probes with high specific activity can be made by. So they can be made by Nick translation and random priming. Answer is option A. PET vector is a low copy number plasmid vector. PET vector has a rope gene. So rope gene, it will keep the number of copies of the plasmid low. Since the PET vector has a rope gene, it is a low copy number plasmid vector. So both 1 and 2 are correct and 2 is the correct explanation of 1. A is the right answer. Then next one. TA cloning is best suited for TAC amplicons. Yes. TAC amplicons means those PCR products. Amplicons are PCR products which are amplified using TAC DNA polymerase. They will add an A at the end of the amplicon. So that will bind to the T of the cloning vector. So TAC polymerases have limited terminal transferase activity, adding A residues at the 3' end of the amplicons. That is why TA cloning is best suited for TAC amplicons. So both A and 1 and 2 are correct and 2 is the correct explanation of 1. Then the geographical indication registry of India is situated at Chennai. So again a question on geographical indication was asked in January 2024. Then next one. Which of the following treaty is for the deposition of microorganism for patent purposes? It is Budapest Treaty. Answer is option B. Statement 1. M. mutein is a vector incapable of replication in Bacillus. Yes, it is having conditional origin of replication, that is called E1, that can function only in E. coli. So it is incapable of replication in Bacillus. M. mutein can be used for systematic insertional inactivation of Bacillus operans. Yes, because it doesn't replicate there, it can be used for that. So both 1 and 2 are correct, and 1 is the correct explanation of 2. Answer is option D. Then which among the following is an endogenous selectable marker gene? GUS is a reporter gene, GFP is a reporter gene, LUX is a reporter gene. What is left out? CAD, that is carbamoyl phosphate synthetase. So it is an endogenous selectable marker gene. Then Carnegie 20. It is based on P elements of Drosophila. It is a vector for P element mediated gene transfer in Drosophila. Then Mullerian mimicry. It is a type of mimicry where one or more species develop similar appearances. Suppose they may be poisonous or dangerous or unpalatable to the predators. Now the predators, they will eat and see only one species, but upon seeing the second species which is imitating the first species, they will not eat the second species. So this will confer what do you call that? Protection to the second species. Now as in the case of monarch and viceroy butterfly, which show Mullerian mimicry. So the monarch and viceroy butterflies, they share bright orange pattern. So once the predator would have tasted the monarch butterfly, but on seeing and would have known that it is not palatable. On seeing viceroy butterfly, which is also similar in appearance to the monarch butterfly, it will think that it is the viceroy butterfly is actually the monarch butterfly, and it will not eat that viceroy butterfly. So viceroy butterfly is protected in indirect way by the monarch butterfly. This is Mullerian mimicry. And this is positive frequency dependent selection. Answer is option A. In this, the common phenotypes have fitness advantage. And in Mullerian mimicry, it means that the more common a mimicry pattern becomes, the more advantageous it is for the species involved. So it is positive frequency dependent selection. Then next one. Which among the following is an example of convergent evolution? New World crane hawk and Old World harrier hawks. That is example. What is convergent evolution? Where organisms that are not closely related evolve similar traits or behaviors. So these crane hawks, that is New World crane hawk, it is found in Central and South America, whereas Old World harrier hawks, they are found in Africa. That means they are from different continents, they are different families also, but they both have legs that bend forward and backward. So this is an example of convergent evolution. Answer is option B. Then next one. Which among the following is an example of library screening through functional complementation? So it involves introducing first what it will require is a host organism that has a specific mutation or a deficiency. Now you introduce a library of genomic DNA fragment to this host organism. Then you screen for the library of DNA fragments in such a way that they have the ability to restore or complement the host function. Okay, examples when I tell you will understand. Identification of yeast His-B gene in oxotrophic E. coli. So E. coli is lacking histidine synthesis. Yeast genomic library introduced into E. coli. Now colonies growing in histidine free media indicate complementation by yeast His3 gene. Okay, now that is an example of functional complementation. Now see identification of DFNB3 deafness gene from Shaker 2 mouse. So mouse embryonic stem cells with DFNB3 mutation are selected. Then human genomic library introduced into the mouse cell. Cells exhibiting restored hearing by complementation with human DFNB3 gene are screened. Okay. And then the human genomic library is screened for functional DFNB3 gene. So C is also answer. Then identification of mammalian transcription factors by using gene knockout yeast strains. So yeast strains with specific transcription factor genes deleted are selected. Mammalian cDNA library is introduced into the yeast. Then colonies growing under selective condition indicate complementation by mammalian transcription factors. So all of the above are example of library screening through functional complementation. Answer is D. Identify the wrong statement. Oligocapping and capture method. Okay. Oligocapping, it is a method that replaces the cap structure of eukaryotic mRNA with synthetic oligonucleotides during full-length cDNA synthesis or cDNA synthesis. That is oligocapping. Now capture method, it is also used for full-length cDNA synthesis. Now what is mispriming in cDNA synthesis? cDNA synthesis is produced from reverse transcriptase. RNA is used for the synthesis of cDNA. Now when the reverse transcriptase initiates cDNA synthesis at an incorrect location, it will result in incorrect cDNAs. That is called as mispriming in cDNA synthesis. Now read the statement. You have to identify the wrong statement. Oligocapping is a procedure for full-length cDNA cloning. Yes. Capture method is a procedure for full-length cDNA cloning. Yes. Oligocapping solves the problem of mispriming in cDNA synthesis. Yes. Capture method solves the problem of mispriming in cDNA synthesis. No. So D is the answer. Then next one. Identify the correct statement. AFLP and RFLP are codominant markers. No. AFLP is dominant, RFLP is codominant. So A is wrong. You have to identify the correct statement. Dominant markers are scored as present and absent states. Yes. They display two phenotypes. That is, if it is present, then it is dominant, and when it is present in homozygous recessive condition, then it is absent. So dominant markers, they are scored as present or absent states. That's correct. Dominant markers will identify heterozygotes. No, they cannot distinguish between homozygote and heterozygote. Only dominant presence can be identified. That is, if it is you are having gene allele capital A small a, then capital A small a, because of the presence of dominant A in that, it will identify the heterozygote, but it will not say it is a heterozygous condition. Okay. So C is incorrect. RAPD is a PCR based codominant marker. RAPD are dominant markers. So the answer here is option B. Dominant markers are scored as present and absent states. Then identify the correct statement. Serum-free media are less prone to contamination by infectious agents. Yes, because it is free of serum. Answer A is correct. Serum containing media ensures reproducibility and product uniformity. No, they introduce variability. Then cells used for transfection experiments are best grown in serum containing media. No. They are often grown in serum-free media because serum containing media, serum will interfere with the transfection. So answer is option A. Now match the following. EST, express sequence tag. It is cDNA. It is used for cDNA cloning. Yes, it is produced from cDNA clones, ESTs. STES is a codominant marker B1. ISSR is a dominant marker C4. And bar gene is a selectable marker D2. So the answer here is option B, A3, B1, C4, D2. Then which of the following is solely a feature of eukaryotic cell? Mesosomes bacteria, polysomes bacteria, cell wall bacteria, mitochondria not present in bacteria. Very simple question. So mitochondria is the answer.